Hello teacher, TA and classmates. We are from the 29th group and today we will present about our dataset, the dataset about breast cancer. The table of contents of our presentation. First, we will introduce our dataset, our research motivation and purpose, the introduction of SDGs that we use, our fishbone diagram, further dataset introduction, the pre-processing and modeling of our dataset, and also the final conclusion. First, we will tell you about our dataset introduction. Our data was obtained by computing patient breast cancer cells from a digitized image of FNA or a biopsy of a breast mass. The biopsy test will provide a set of data that classify whether the patient cancer cells are malignant or benign. There are 569 instances of our data and 32 attributes. Our dataset is donated to the University of Wisconsin Computer Science Department in 1995. In this slide, we attach the attribute information of our dataset, which consists of diagnosis, which is the target of our dataset, which have two variables, malignant and benign. Second one, we have radius, which is the mean of distances from center to point on the perimeter. The texture is the standard deviation of grayscale values, smoothness, local variation in radius length, compactness, concavity points, fractal dimension, and so on. Each of these attributes have three variations, which is the standard error and worst or largest. Standard error is the statistical accuracy of the sample's mean, and worst or largest is the mean of the three largest cancer cells. Our research motivation and purpose is to give further knowledge of breast cancer, to raise people awareness of breast cancer, and to provide a machine that can predict one's breast cancer, whether it's malignant or benign. Third, we will go to the introduction of SDG that we use. Our dataset is based on the third SDG, which is about the good health and well-being. The main idea of the third SDG is to ensure healthy life and promote well-being for all ages. The target of the third SDG is to cover and focus on various aspects of healthy life and healthy lifestyle. There are 30 targets, and the progress towards the target is measured using 28 indicators. Breast cancer is included in the fourth target, which is ensuring reduction mortality from non-communicable diseases or NCD and promote mental health. Why? Because breast cancer is categorized as a non-communicable disease. It's a disease that doesn't spread through people. Instead, it's a disease that are associated with somatic mutation in breast cells that are acquired during a person's lifetime, and also occurs as a result of inheriting genes that contain certain mutations. The third SDG aimed at by 2030 reduce by 1/3 premature mortality from NCD through prevention and treatment, promotion about mental health and well-being. Why is the fourth target important? It's because NCDs are the leading cause of death globally. In 2012 only, NCDs caused 68% of all deaths, which is 38 million, up from 60% in 2000. How come NCDs are preventable? NCDs are preventable because the cause of this disease rely on the person's background, lifestyle, and environment. The fourth content of our presentation is about our fishbone diagram. In this slide, we attach the fishbone diagram of our dataset, and our main idea for the fishbone diagram is how is breast cell nuclei affected by breast cancer. The fishbone diagram have three major factors, which is the dimension, surface, and density. And for the dimension factor, there are three middle or minor factors, which is the radius, perimeter, and area. For example, radius size of a breast cell on someone will be affected if the person suffer from breast cancer. And the next one is surface, which is the minor factor are texture, smoothness, concavity, and concave points. And for the density factor, there is the compactness minor factor. Our next content is the further dataset introduction, which we will check the data value and missing value of our dataset. We will be using Google Collaboratory software for this project. And first of all, we will need to import all the necessary items needed. We will import pandas, numpy, seaborn, and a few items from sklearn. And after all is done, we can move on to the next one, which is reading or loading our dataset. into the Google Collaboratory software. Here we are displaying the 5 first instances of our dataset, and as we can see, all of the data is already in a table that can later be used in the next part. Then we will be executing the df.shapeline, which will give us the information of how many columns and rows is there on our dataset. And as we can see, there are 569 rows and 32 columns, which is correct as we are introduced before, 569 instances and 32 attributes. Next, we will be checking our dataset information, the attribute information. And as we can see, most of the attributes that will be used in the next part are all floats, and because all of them are numerical values, we can directly use them in the next part because of all of the numbers are decimals already. And then using the df.isna, we can check whether our attributes have missing values. And as we can see here, there are no missing values. All of our instances are filled in completely. Then we will be using the describe method to display all of the columns on our dataset. The first one, there is count line, which is the number of non-empty values. And as you can see, all of the columns have 569 non-empty values, which is correct, no missing values on each column. And it will also display the mean, which is the average value of each column, also the standard deviation, minimum and maximum value. The first quartile, middle quartile, and third quartile will also be displayed here on the 25%, 50%, and 75% row. Next, we will be using label encoder to convert the diagnosis column, which initially consists of M and B strings, into the number 1 and 0, so that it can fit into our data and into the machine learning model. The number 1 means the diagnosis is malignant, and the number 0 means the diagnosis is benign. Next, we will be using histogram and pie chart to display the total number of our instance. And as we can see on the histogram part, the left side, there are roughly 357 cases of benign breast cancer on our dataset and 212 malignant breast cancer case in our dataset. On the right side, as we can see on the pie chart, 62.7% of our instance is the benign breast cancer case, and 37.3% of our data set is the malignant case of breast cancer. Next, we will be using the matplotlib to display the correlation matrix of our dataset. The matrix is titled breast cancer attributes correlation, and the dimension is 32 times 32, which is the number of our attributes. And the matrix will show the correlation of each features or each attributes. From the output graph below, the red colors mean that the attributes are positively correlated with each other. The red colors is the number 1, and blue is the number 0, which is number 1 is the positive correlation and blue is the negative correlation. The yellow and green patches suggest that there are some moderate correlation, and the blue one, as I just said just now, have negative correlation. This matrix is good to check the correlation between each attribute, so we included it in our data introduction. Next, we will be putting the diagnosis or our dataset target column into a separate data frame, so we can split our dataset into train and test data. After putting the diagnosis column in a different data frame, we will be dropping the diagnosis column and ID column because the ID column is not relevant into the next process, so we can drop it. And here, as we can see, we display 5 of the first instances, and the ID and diagnosis column has been dropped. So good. Next, I will be presenting about the pre-processing process of our dataset, which includes the min-max scaler and PCA. So first we will be transforming our data into the NumPy array. Next, we will proceed to the min-max scaling process. So scaling means to change the range of values, but without changing the shape of distribution. And the min-max scaler subtract the minimum value in the future and then divides it by the range. And the range is the difference between the original maximum and the original minimum. The min-max scaler also preserves the shape of the original distribution. It doesn't meaningfully change the information embedded into the original data. The range of the attribute returned by the min-max scaler will be in the range of 0 to 1, as we can see in our table here. Next. So the PCA process we use to our dataset is to reduce the dimension in machine learning, because high dimension means that the dataset has a large number of features or attributes, and many algorithms that work fine in low dimensions become intractable when the input is in high dimensional. So the PCA will improve the model performance in most scenarios. It basically compress a lot of attributes into smaller ones decided by the n components used here. We use the n component 0.99, and after running it, the PCA will compress it into 2 attributes only. We actually already tried to use the n components of 0.95, but result is much better if we use the variable 0.99. So after the PCA, we hope that the modeling process is smoother and hopefully more accurate. We will see in the final conclusion later whether the theory is correct towards our database. And the next content of our presentation is about our dataset modeling, which will consist of decision tree, RFC, naive Bayes, and KNN modeling process. So before continuing to the modeling part I just mentioned, we will split all three data, the original, scaled, and PCA data into train and test data. Our dataset will now then consist of 80% train data and 20% test data, which as shown on the line test underscore size 0.2. The train data will be used by the machine learning to learn, and the test data will later be used to test the accuracy of the machine. After splitting, as we can see, all of the three data, the original, scaled, and PCA are all the same. The train data is 455 instances, and the test data is 114 instances, which all have same 30 columns. The three splitted data will later undergo the decision tree, random forest classifier, k nearest neighbor, and naive Bayes process, and later we will find the final conclusion. Now let's move on to the decision tree modeling. The decision tree will use classification and regression tree algorithm, which is simply as a question, and based on the answer yes or no, it will further split the tree into subtrees. There are internal nodes that represent the features of the dataset, branches that represent the decision rules, and each leaf node represents the outcome. There are two nodes, which are the decision node and the leaf node. Decision nodes will be used to make any decisions and have multiple branches, whereas leaf nodes are the output of those decisions and do not contain any further branches. Using pre-processed and splitted data, information gain and entropy will be executed to decide what attribute will be put in which node. And here we executed to all three types of data, and as we can see here, the F1 score is the accuracy of the three data, and on the last part, on the last picture, the conclusion one, it will be plotted here, so we can compare the three accuracy. And as you can see, using the original data, decision tree modeling will have the highest accuracy of roughly 94.8%. So as we can see here in this slide, I will attach the decision tree of our dataset that we obtained after running it. On the top, we can see the root node. It is where the decision tree starts. It contains the most information, and it represents the entire dataset, which further will be divided into two or more homogeneous sets. Then below we can see the branches. That is a tree that formed by splitting the tree. And we also can see a lot of leaf nodes, which is the final output node, and the tree cannot be segregated further after getting a leaf node. Next one is the random forest classifier. It is a classifier that contains a number of decision trees and various subsets of the given dataset and takes the average to improve the predictive accuracy of that dataset. So instead of relying on one decision tree, the random forest will take the prediction from each tree, and based on the majority votes of predictions, it will predict the final output. The greater the number of trees in the forest will lead to the higher accuracy and prevents the problem of overfitting. After consideration, we will use the n estimator of 1000, which will create 1000 trees. And basically, the step of the random forest is they will select random k data points from the training set and build the decision trees associated with the selected data points or subsets, and then repeat it the same process 1000 times, and finally we have the random forest. So the same, we also apply it to the tree data here, and as you can see, the original data which are modeled using the random forest will have the highest accuracy of roughly around 97.5%. So for the next modeling process is the k-nearest neighbor modeling. The KNN algorithm assumes that the similarity between the new case or data and available case, and put the newest case into the category that is most similar to the available ones. The KNN algorithm stores all the available data and classifies it into a new data point based on the similarity. For our database, the KNN will find the similar features of the new breast cancer data and then set it to the malignant or benign case based on the most similar attribute. Then it will be put in either the malignant or benign category. Here we use the component neighbor equals to 5 so that the machine will look at the 5 nearest neighbor and decide whether the attribute is the same, and will be put it into the category the same as the major neighbor category. And as we can see here, the scaled KNN has the highest accuracy of 97%. Okay, so for the last modeling process is the naive Bayes. Naive Bayes is a probabilistic classifier which it will predict on the basis of the probability of an object. First, it will convert the given dataset into frequency tables and generate the likelihood table by finding the probabilities of given features. And now, using the Bayes theorem, it will calculate the posterior probability, the same as that we learned in the class homeworks. And for this naive Bayes model, we use the Gaussian model because we assume that the features follow a normal distribution, and it's the best using Gaussian model. Meanwhile, because the multinomial naive Bayes classifier is used when the data usually is multinomial distributed, and the Bernoulli usually is used when the variables are the independent Boolean values. So for our dataset, we decided the Gaussian model is the best one. And as we can see here, after we run the data, we got the accuracy, and we compared it at the bar chart below. As you can see, the scaled data have the highest accuracy of 96%. So finally, we come to the final conclusion of our presentation. In this slide, I will attach the program to plot a bar so that we can compare all of the modeling process that we have done. And later in the bottom, as we can see, there are 4 charts. It is obtained by running the program 4 times. We actually run it many times, but we only attach 4. And as we can see, each time we run the program, it will have different result, different accuracy on each model. And after comparing it, we can see that majority of the nodes is highest on the scaled data KNN modeling part, which have roughly, after I calculated it, is around 87%. So by looking here, finally I can conclude that For this data set, to see whether someone's breast cancer is malignant or benign, we can use the KNN modeling using the scaled data to receive the highest accuracy possible. And yeah, that is my conclusion for this presentation. Here is the end of our presentation. Thank you for listening.